Hello, my name's Larry Harris. I was a professor of zoology and then later biological sciences at UNH for many years. Back in 1970-71, I was an assistant professor in the zoology department. And I was approached by an ocean engineering professor, Godfrey Savage, who was head of EDAL, and he proposed the idea of a study on the possibility of introducing the Dungeness crab from the West Coast to the Gulf of Maine as a supplemental fishery to lobsters. And the basic idea was to conduct a study out at the Isles of Shoals using the EDALHAB, which was built by ocean engineering students a couple of years previously. This is a movable underwater habitat, and there would be 2 zoology students and 2 ocean engineering students as aquanauts. And I thought this was a really interesting idea, so I reached out to some of the students who were in the zoology department, and we put together a team. 2 students were selected, Tom Glennon and Eric Sawtell, to be the aquanauts.
My name is Eric Sawtell. I was a zoology student at UNH and graduated with a BA in zoology. Zoology was actually in the College of Liberal Arts back then. And I was in the class of 71, 72.
My name is Tom Glennon. Back in the spring of 1971, I was an undergraduate zoology student in my senior year, and I was looking for an elective course to take to round out my curriculum and graduate that June.
And that Ocean Projects course was under the direction of Dr. Larry Harris, who had only been at UNH a couple of years, and in the zoology department, and then Dr. Godfrey Savage, who was the director of the engineering design and analysis laboratory. You had a bunch of kids really under the tutelage of 2 really forward-thinking people that in Larry Harris and Jeff Savage, who sort of mentored us to get us through the process of how is this thing going to work.
And I came across a special projects course in the zoology department, which piqued my interest. And I joined the course, or signed up, not knowing that it would develop into probably one of the more exciting projects in my lifetime.
Dr. Savage had the idea of developing a mobile underwater habitat, which could be attached to a surface vehicle and moved around and moored to the bottom, which would be essentially a living platform or a living habitat for divers that were doing extended periods of research underwater. And then after an extended period, they would come to the surface only one time and enter a decompression chamber and slowly be brought back up to atmospheric pressure without having to go through the process of having the bends.
To assist us, they were Barry Walden, Bob Ballard, and Bill Rainey from the Deep Submergence group. And Woodsole also let us utilize their tender called the RV Lulu, which normally would support the deep submersible operations of the Alvin.
And a second vessel, the RV Undaunted from the from the Cape Fear Technical Institute, was used as the base for the team of zoology students who were doing the support diving. We then shipped a group of adult male Dungeness crabs from the West Coast, and we then kept them in Spalding Hall in a recirculating system. The team of students conducted a series of studies, feeding behavior, or feeding preferences, and behavioral interactions between these crabs and some lobsters that we had purchased. And a large cage, 12 feet by 24 or 36 feet and 4 feet high, was deployed on a sandy bottom next to the EDALHAB. Eric and Tom and the two ocean engineering students were deployed to the EDALHAB and spent 4 days and a series of dives monitoring the interactions of crabs and lobsters that were put into the cage, making observations on behavior and such.
Our responsibility was to make these periodic trips and go along the cage and just record the interaction between some West Coast crabs called the Dungeness, quite large crabs, and how they would react to the ambient water temperatures, plus some of the native species of crustaceans and bottom feeding finfish in the cage itself.
So we were diving on a rotational basis with a surface diver coming down to assist us in doing observational recordings of how these Dungeness crabs and other indigenous crustaceans such as lobsters and crabs and some finfish all interacted in this caged-in unit that we had. And so we did dives every 20 minutes, 24 hours a day, every 2 hours, 20-minute dives, 24 hours a day for 4 days.
Later, we retrieved the crabs and lobsters, and also some collected scallops, and had a seafood feast to celebrate the system. Have to realize that back in 1970, this invasive species were not a major issue. In fact, New Hampshire Fish and Game was actually introducing coho salmon to the Great Bay Estuary, something that wouldn't be considered these days, but at that time it was a totally different game. The final decision was it wasn't really a good idea to introduce a new crab to the Gulf of Maine, and so it was a pretty amazing experience.
The project involved refurbishing the habitat that was originally built in 1968 by some engineering students here at UNH. And then 3 years later, we utilized this for EDALHAB 2 project, despite the fact that it was pretty low budget and done by a bunch of engineering students, and it was very successful. And to this day, EDALHAB remains as one of the most mobile underwater habitat systems ever. It's a good, I guess, exercise in adapting something and innovating and making something work with what you've got.
The habitat that was utilized for the program was first developed and built by a group of engineering students in the EDAL section of engineering. It was built in 1968. It was a converted pickling vessel. That was actually used in the 1968 experiment in Alton Bay, New Hampshire, which is part of Lake Winnipesaukee and the Lakes region of New Hampshire. The habitat itself was situated on the Port Authority Pier in Portsmouth, New Hampshire, had to be raised and lowered into the water by a good-sized crane, and then directed between the two hulls of the Lulu and raised on an underwater platform that the Alvin typically used, and actually steamed out to a location a few 100 yards off Appledore Island at the Isles of Shoals in the Gulf of Maine. The habitat was lowered and submerged between the hulls of the Lulu and was secured in about 50 feet of water on a sandy bottom by members of the world famous Boston Sea Rovers Diving Club, who also acted as support divers during the project.
So this particular EDALHAB project, and EDALHAB stemmed from the name of the Engineering Design and Analysis Laboratory, shortened, and as an underwater habitat, which was at the time really in the genesis of saturation diving research, which allowed divers to stay at a certain depth for an extended amount of time under saturation, which is where the diver's body, tissue, and blood reaches the nitrogen gas in the body reaches equilibrium with the gas that they're breathing in their lungs. And normally, if a diver was coming from the surface, they could only stay at depths below 33 feet, which is 1 atmosphere pressure, for a short amount of time before having to resurface to rid their blood and tissues of nitrogen, excess nitrogen, which if they didn't do that, they'd result in nitrogen saturation, or what's called the bends, where the gases would essentially come out of solution, just like opening a soda bottle, and carbon dioxide bubbles. And at that time, there were 3 other habitats at least. The Navy had a Sea Lab 2 project, which was a stable underwater habitat. And then Tektite, which was another major project. And then there was one in Germany called Helgoland. Some of those, unfortunately, had some diver deaths associated with them. And they were not mobile, they were stationary.
The first evening of the mission, there were 3 saturation divers, including myself. The others were Eric Sawtell, who was my course mate in zoology, and Mark Hurdle, who was in the mechanical engineering department at UNH. So I remember very vividly the 3 of us diving in Blackwater. It was in the evening. We were told to just follow the lights of the habitat while it was at the bottom.
We, as the saturation divers, had to wear red suits so we could be distinguished from the surface divers in case something happened. Another funny part of it was that we drew straws to see who was going to be the first one to go into the habitat. Tom Glennon got the short straw. And he said afterwards that he was a little in trepidation of what when he took his mask off inside the habitat, what was his first breath going to be.
There was a little intrepidation in taking that first breath within the habitat, but I managed to take my mask off and took a breath, and it seemed fine. So we all 3 of us entered. So the responsibility of the 3 of us was that Eric and I would take periodic dives into the environment and be supported by a surface diver, normally one of our zoology comrades.
If you can imagine, the accommodations inside the habitat were not exactly the Four Seasons Hotel. It was hot because we had to have a heater in there because the water temperature was right around, not quite 50 degrees. Even though the habitat was insulated, you could not sleep inside the habitat. There were 2 bunks in there, but the amount of noise, the humidity, taking off a wetsuit and putting on a wetsuit was no fun after 4 days, let me tell you, but we did it.
And I have to say, it was a small space that consisted of a folding card table, 3 chairs, and a bunk with 2 beds, which we never used. We never slept. The inside of the habitat was very loud with those bubbles going along the outside. The air coming into the chamber came through a valve, which was very loud. The humidity was so high, unfortunately, the steel ceiling of the habitat dripped constantly down on us and totally saturated the bedclothes almost immediately, preventing us from getting any sleep at all.
The food was actually came from the surface in pressure cookers because there was no other way to get food to us. Usually by the time we got it, though, it was cold.
The only highlight really that I remember was the fact that our meals were sent down to us in pressure cookers. The meals were cooked on board, I believe on the Undaunted, from what I understand, sent down to us again by support divers, and we would take them, open them up, and luckily everything stayed dry and the food was delicious.
The last night we were there, the way the habitat was set up on one end of and this was a small chamber. It was 8 by 12 chamber that we were living in, and one end had 2 bunk beds in it, along the side, and we had to sit over what we call the moon well, which was essentially a 275-gallon oil tank welded to the bottom of the habitat. We came up through that on a ladder to get in. Well, that had a piece of plywood over it for a door, and we had to set a card table over the top of that in order to sit there and eat or do whatever work we were doing. So the last night we were there, we were pretty well cooked by that time, and the chef on the Lulu sent down a roast pork dinner for us. Well, last night we were there before we were coming up to go into the decompression chamber, and we had it all set up on the table, and everybody was ready to dive in. And I won't mention who it was, but one of the other divers happened to hit the leg on the card table, and in very slow motion, everything slid off and right down in the well because we hadn't put the cover over it. And so by this time it was, I don't know, 7:30 at night, and everybody topside had already finished. The cooks had finished and everything. We sent down 3 peanut butter sandwiches for us.
Project wrapped up, we had to be concerned that being saturated under pressurized conditions, the nitrogen we were breathing both in the habitat and in the tanks we used when we went out underwater was creating a saturation of nitrogen on our bloodstream. And you have to be very careful coming up to the surface, because if you do it too quickly, the nitrogen becomes gas and is released into the body and can become concentrated in your joints and cause a condition called the bends.
The decompression chamber was even worse because it was essentially a 3-foot diameter chamber that we were in for 11 hours. You couldn't move around much in it. And
climb up the ladder where everything was stripped off of us except our suits. I believe we went in with our wetsuits, and then we took them off in what we call the Yellow Banana, which was a decompression chamber, which was managed by the medical officer of the project, Dr. Joe McGinnis.
I already had my suit on, but they were putting my tank on and making sure everything was all set. And one of those things was to put a weight belt on me. Well, they didn't put the weight belt on. So I jumped in and I kept bobbing like a cork because these suits were so buoyant that I just couldn't get down. And so after about 3 attempts to try to get below the surface, I popped back up and I said, which one of you guys dressed me? And they were all laughing because they knew I didn't have my weight belt on.
I do remember one very pleasant event was we were able to finally shower after 4 days. I believe the showers are on the Lulu project. I believe was successful and had a lot to do with the university becoming a Sea Grant university.
So the Sea Grant programs recognized that UNH was really in the beginnings of marine and estuarine research, and to this day, I think we have shown that we're right up there with the best. Actually, this project and a couple of other projects, one other project that I was involved in, were really the genesis of UNH becoming a Sea Grant University in 1980. I think the important thing was that they taught us how to look at the big picture, and that has carried with me throughout my whole life.
I think the rest is pretty much history, and it's amazing that 50 years later, we're now celebrating this interesting event, and you can visit the EDALHAB at the Seacoast Science Center in Rye.